Welcome everyone, and thank you for joining today's webinar on consumer product biocompatibility testing. Our presenters today are Mary Dothage, Senior Manager, and Dr. Nick Allmeyer, Manager of Analytical Chemistry with EAG Laboratories. I'm Melissa Campbell, Marketing Manager with EAG, assisting with today's event. So before we get started, just a couple of housekeeping items. All attendees are muted. However, if you have questions during the presentation, you can enter them in the control panel or app, and we will address them during the Q&A session. At the end of the webinar, a survey will pop up on your screen, and we would really appreciate your feedback. And now I will turn it over to Mary.
Thanks, Melissa. Good afternoon. It's a pleasure to be speaking with everyone today. Thank you for taking the time to join EAG's webinar, Consumer Product Biocompatibility Testing. Wearables, virtual reality equipment, and electronic cigarettes are all examples of products that come in direct contact with the user. With any device in contact with the consumer, there are safety questions we may ask ourselves. These questions include, are materials of construction suitable? Is a compound of concern present? At what level is a compound of concern present? Is manufacturing of the end product consistent? Is a recall required? Essentially, is the product safe for consumers? If a product is found to be unsafe for the consumer, the loss of revenue for a company can be manifested in multiple ways, including customer lawsuits, product recalls, and unwanted regulatory agency attention. In the webinar today, my colleague, Dr. Nick Allmeyer, and I will discuss EAG's approach to consumer product safety testing. In addition, we will offer case studies to highlight unique customer requests with catered testing solutions. You may be aware of a term, biocompatibility, that's used to measure consumer safety. But in case you're not familiar with it, let's go ahead and define it. Biocompatibility is defined as the ability of a material to perform in a specific application without causing an adverse reaction in the body. The U.S. Food and Drug Administration, or FDA, mandates biocompatibility must be evaluated for any medical device that comes in direct or indirect contact with the human body. Because of this mandate, the FDA provides guidance on how to test for biocompatibility through test standards like ISO 10993. ISO 10993 provides clear guidance on testing procedures to evaluate biocompatibility of medical devices. But we're not here today to speak about medical device testing. How can we assess biocompatibility in consumer products? Consumer products, like medical device, come into direct contact with the consumers. However, consumer products lack regulatory guidance on how to measure product safety. Some test standards are being drafted at the request of the Consumer Product Safety Commission, or CPSC. For example, Underwriter Laboratories is drafting UL 8400, which provides safety testing for virtual and augmented reality equipment. However, these test standards are less specific than medical device regulation in respect to testing guidance. Other consumer product test standards have a narrow scope and do not assess the complete biocompatibility of a device. For example, EN 1811 denotes safety testing centered around nickel release in jewelry. This is a narrow scope which assesses nickel release of the jewelry but would not identify other compounds of concern. So how do we design studies to assess biocompatibility and ensure user safety in consumer products? The truth is, it all starts with a conversation. Product information, including device composition and device use, are necessary to custom design a biocompatibility experiment. Biocompatibility is commonly assessed using extractable leachable studies. What are extractable leachable studies? Extractable leachable studies identify chemicals that have the potential to migrate from a product. These studies are twofold in nature. The first part, the extractable studies, are used to identify compounds of concern the consumer may be exposed to or a worst-case scenario. Leachable studies are designed to monitor the substances the consumer will be exposed to during normal use. Leachable studies are more representative of everyday use. Each extractable leachable study should be designed with the device use in mind. EAG takes several steps to design a study like this. We have summarized the cycle of an extractable leachable study in this graph. As we have discussed, product information including device composition and device use are necessary to custom design an extractable leachable experiment. This ensures the study is representative of the device use. Samples will be analyzed to identify chemical compounds leached from the device. The identified compounds will undergo a toxicological review to establish compounds of concern that are present. The concentration of the compounds of concern will be determined to finalize the risk assessment. Based on the risk assessment, a safety decision will be made. All information obtained from the study then gives additional information about the product. The product information gained from the study can help make decisions about final product design. There are many manufacturing steps to obtain a finished product. Product manufacturing begins with raw materials, which are molded into modular components. The modular components are then assembled into finished product. Many base polymers, plasticizers, UV stabilizers, pigments, adhesives, or antimicrobials can contain compounds of concern. It is important to know the device composition for two reasons. One is to know what chemical compounds may be present so we can screen for them. The second is to ensure the method of analysis is suitable to detect the compounds of interest. After we have established the device composition, the next step is to discuss how the product is being used. Important pieces of information include: what is the duration of time the device is in use? What are the conditions of device use, including temperature, humidity, or pressure conditions? Information about product use determines the extraction conditions for that product. For example, a device that is used for 2 hours will be extracted differently than something that is used continually. We can assess compounds of concern that may come in contact with the consumer by testing with mediums such as artificial sweat or artificial saliva. A fitness tracker would not be leached in the same medium as a musical instrument mouthpiece. Finally, before any study, we make sure our proposed biocompatibility assessment is aligned with established test methods. Remember the UL 8400 and EN 1811 methods I mentioned earlier in the presentation? We can modify the principles and established test methods to use as reference to custom design a biocompatibility experiment. Let the guidance guide us. I'll now turn the floor over to my colleague, Dr. Mick Allmeyer, to discuss EAG capabilities as well as some case studies.
Thank you, Mary. EAG has partnered with clients for a variety of samples and testing opportunities, including finished products, components, and raw materials. Testing can include several categories of studies: biocompatibility, restrictive substance, and investigative. Biocompatibility studies consist of extractable leachable, off-gassing, and in vitro testing, all of which we can support at our lab in St. Louis. Restrictive substance studies are focused on exclusion list testing to rule out certain chemicals of concern that are at specified levels that are dictated by regulatory agencies. Investigative studies are experiments performed to solve critical issues to find out what happened. These studies often take place after the first two testing categories don't turn out as expected to determine a root cause. EAG is skilled in non-routine investigative testing, and we have a well-equipped lab that is staffed with highly educated scientists to solve these issues for our clients. To facilitate these studies, there are three primary types of instrumentation we rely on that I'll outline next. The first is liquid chromatography mass spectrometry, or LCMS. This instrumentation relies on liquid mobile phase to separate compounds before a mass spec detector with ESI or APCI ionization. Typically, semi-volatile or non-volatile chemicals are detected by LCMS. EAG has 8 thermo Orbitrap high-resolution accurate mass instruments. We first started with the Q Exactive that we purchased about 10 years ago, and more recently we've acquired 2 Exploris 120s, 1 IDX tribrid, and 4 IQX tribrid mass spectrometers. These Orbitraps are high resolution with excellent mass accuracy. Typically, the resolving power of an Orbitrap is several times higher than what can be attained by a TOF instrument. Our tribrid instruments are primarily utilized for biocompatibility testing, and they have a resolution of 500,000. Additionally, these instruments are capable of collecting a plethora of MS-MS fragmentation data. Equipped with a quadrupole, a linear ion trap, and an Orbitrap detector, spectra can be collected from MS2 up to MS10 with both HCD and CID activation to aid in our structural identifications. The second primary type of instrumentation we use is gas chromatography mass spectrometry, or GCMS. This instrumentation utilizes gas as the mobile phase to separate compounds before a mass spec detector with EI ionization. Typically, volatile and semi-volatile chemicals are detected by GCMS. EAG has more than 20 GCMS instruments in St. Louis alone. These are primarily comprised of single quad instruments, but we also have 2 GC-QTOF high-resolution instruments that can aid in solving for unknowns by using both normal and low-energy EI ionization. This allows us to observe the molecular ion and fragment ions in spectra. Additionally, our GCMS instruments are capable of other methods of sampling. Direct injection of extraction solutions is the most common, but alternatively, we can perform heated headspace sampling to analyze for volatile chemicals. In this case, we heat the sample in a sealed vial, then draw from the headspace where volatile analytes are present in equilibrium and inject them into the GC. The third primary type of instrumentation we use is inductively coupled plasma mass spectrometry, or ICP-MS. ICP-MS is used to determine the concentrations of elements in a sample by ionizing the sample with a high temperature plasma and then detecting the ions with a mass spec detector. Typically, metals are the focus of ICP analyses, with nickel being mostly of interest for skin sensitization, but other metals such as chromium can be an issue depending on the form. So what do we do once we've acquired all this data? Well, now I'll focus on our screening capabilities at EAG. EAG is capable of analyzing extracts against a threshold limit, such as an analytical evaluation threshold, or AET, like an ISO 10993 Part 18 study. But we're also capable of performing exclusion testing for certain types of chemicals, like sensitizers, and we can do this at a much quicker turnaround time than a full Part 18 study. To process LCMS data, we utilize Compound Discover for data deconvolution, database searching, and then annotation of the results. MS-MS spectra are searched against the ThermoMZ Cloud library containing spectra for more than 33,000 compounds. And we also have analyzed over 1,000 standards that we've added to our proprietary database with a focus on extractable leachable compounds and sensitizers. For focus screening purposes, we've also built a sensitizer mass list based off a combination of the European Chemicals Agency CNL inventory, compounds that we've also added by analogy through SciFinder substructure searches, such as acrylates and glycidyl ethers, and from other thermal mass lists. All this software requires a lot of computing power, and to support this, we've built more than 10 gaming-style PCs, like this one in the bottom right of the slide. Our scientists are experts in using the software to interpret the data and come up with good annotations. And similar to LCMS, to process GCMS data, we use other software, such as MassHunter, the NIST library, and our internal database. MassHunter Qualitative Navigator and Unknowns Analysis are used to integrate peaks, search spectra, and deconvolve overlapping peaks into individual components. So, for example, you may have, like in the bottom of the slide, something that appears to be one peak, but it's actually composed of 3 peaks hidden within one. Using MassHunter Unknowns Analysis, we can deconvolve this one peak into 3 individual components. Spectra are then searched against the NIST library and commercially available databases. There's over 347,000 chemical compounds in the NIST library, and this covers a large number of chemicals that one might detect. However, it's not comprehensive, so we've also built our own library focused on sensitizers and polymeric fragments that supplement this library that's available. So next, I've chosen some case studies to present that go beyond typical biocompatibility testing. These case studies highlight our capabilities and demonstrate the ways we can help customers determine the risk associated with their products and also troubleshoot manufacturing issues. This first case study involved differentiating between two regioisomers of bromo 2,4-dinitroaniline. The backstory on this problem starts with the extraction of a product composed of dyed black materials. Fabrics and pads are often dyed with a mixture of colors to produce black, such as orange, blue, and violet. Just to give an example, one of Dystar's patents specifies the use of a mixture of dispersed orange 288, dispersed blue 291, and dispersed violet 93 to dye polyester fibers black. These dyes can also be referred to as azo dyes, given that they all contain an azo functional group. Unfortunately, azo dyes are known to degrade into amine byproducts through cleavage of the azo bond. This cleavage can take place even if the dye has been effectively fixed to the fabric. So in this case, the dyes were cleaved to give bromo 2,4-dinitroaniline in the sample extracts that we observed. Now, bromo 2,4-dinitroaniline can exist as several regioisomers. We weren't able to confirm by comparison to the NIST GCMS library whether the bromo 2,4-dinitroaniline that we detected was actually the 6-bromo isomer or the 5-bromo isomer shown in the bottom left of the slide. As you can see where the arrows are drawn, the 5-bromo isomer has a bromine that's ortho to the nitro group. This makes it a good electrophile and a potent sensitizer as a result. This had toxicologists concerned about the risk associated with the samples we had tested. And to make matters worse, 5-bromo 2,4-dinitroaniline is not commercially available. But luckily, we have synthesis capabilities in our lab, and we quickly identified that we could prepare a reference material starting with 1,5-dibromo 2,4-dinitrobenzene and reacting it with ammonium hydroxide to form the reference compound. After preparation of the 5-bromo 2,4-dinitroaniline, we used NMR to unambiguously show that we had prepared the compound of interest. Looking at the spin-spin coupling, the top spectrum for the 5-bromo shows only singlets since the protons are too far apart and they're separated by 5 bonds. For the 6-bromo spectrum on the bottom, metacoupling is observed because these protons are only separated by 4 bonds. So as you can see, we spectroscopically confirmed which isomer we had prepared as a reference material, and we had a suitable standard for other testing. We took these standards and compared their fragmentation by LCMS using both the CID and HCD activation that I mentioned earlier. We observed a difference in ion abundances and unique fragment ions for each regioisomer in our MS2 spectra. This would have been difficult to theoretically predict, but this allowed us to confirm that the safer regioisomer, the 6-bromo, was present in the samples we tested, and then we were also able to add this spectra to our database for later assignments by LCMS. Moving on to the next case study, we have chemical migration testing. So testing for the migration of chemicals from food contact materials is well known, and it's an area that's subject to regulations. But currently, testing for the migration of chemicals in consumer products is not as common. Volatile and semi-volatile organic compounds can off-gas or migrate from the internal electronics of a device to the user-facing soft good. To test for this, samples are heated in a sealed environment, such as their packaging or gas sampling bags. This can simulate long-term storage, such as sitting on a shelf waiting to be purchased, or to simulate the heat that they generate when they're being used. The soft good is then extracted to test for compounds such as isobornyl acrylate, or IBOA, which is a potent sensitizer that's used as an adhesive in electronics. Some examples of the types of soft good components that can be tested are the padding in headphones, or maybe the nose pads for smart glasses. And these devices can be broken down into functional components and tested individually if a root cause analysis is needed to determine where the chemical of concern is coming from. Other off-gassing experiments that we've performed are more focused on odor samples or potential residual chemicals. Everyone's likely experienced opening a new product and noticing kind of an unusual chemical smell at one time or another. The source of these smells can be typical residual chemicals from manufacturing, or they can be something worse that's due to some issue with the manufacturing. Solid phase microextraction, or SPME, can be used to test for these chemical smells. In this case, the samples are incubated in a gas sampling bag, and SPME fibers are exposed to the bag for an additional period of time, then analyzed by GCMS. So in the bottom left, I have a general schematic for how the fiber would be exposed to a sample and then desorbed on the instrument. But for consumer products, these would typically be done in a gas sampling bag, like the ones in the bottom right here. These bags can be as large as 100 liters or smaller than 1 liter, depending on what the size of the sample is and what's needed. The next case study I have is focused on the detection of isocyanates and the development of methods to determine if these detections are real to the sample or the result of thermal degradation on the inlet. Thermal degradation can occur during GC analyses both at the inlet or on the column. The top right chromatogram shows a general example. In a perfect world, the parent peak would be observed with no degradation. But if the compound degrades on the inlet, it could split into 2 or more peaks, shown as front-end degradation or labeled as A on this chromatogram. And you would never see the parent compound that's actually truly in the sample. Another example would be if there's on-column degradation, which would be the chromatogram B. This would give you a broad peak that also would not render the parent compound on the mass spectrum. It's known that polyurethane linkages, such as those that are used in polyurethane foam, can thermally degrade and give isocyanates such as MDI, like I have in the bottom left of the slide. To detect when this is happening, we use something different called a multimode inlet, or MMI. This is capable of low temperature injections that can gently introduce the sample onto the column rather than introducing the sample to an inlet that's hot at maybe 270 degrees. The inlet temperature starts off low, and then gradually increases to the target temperature over time, like I have shown in this inlet program in the bottom right of the slide. This allows us to reduce and eliminate inlet degradation to isocyanates and also observe a retention time delay from a standard that can tell us that degradation has in fact occurred. The final case study I have to present involves a similar issue related to thermal degradation. So phenolic resins are commonly used in consumer products, and residual phenol can be a sign of poor curing of those resins, or can also be of toxicological concern if the levels are high enough. We found high levels of phenol in samples that were tested by GCMS. To confirm this finding, we developed a UPLC FLD method that relies on fluorescence to evaluate phenol in a non-thermal manner. Just for an example, I have a chromatogram shown of a phenol calibration curve stacked, showing how good of signal we get in linearity. In the end, for this particular problem, we were able to identify that the phenol levels were quite low in the sample, and that it was in fact thermal degradation of phosphorus-based flame retardants such as phenyl phosphate that were giving the high levels of phenol detected in the GC. So this wraps up the case studies that I've prepared. And in conclusion, there are many steps in a product lifetime where safety issues can arise. So how can EAG help? EAG Laboratories is an independent testing lab which can provide unbiased results for manufacturing audits, raw material examination, product design, finished product examination, and retail product quality control. We have years of experience testing consumer products, including VR equipment, wearables, electronic cigarettes, exercise equipment, children's toys, and more. So thank you for your time today, and I hope we provided some insight on how to alleviate concerns related to safety for consumer products. If there are any questions, we'd be happy to answer them. And please submit your questions in the question panel located on the bottom right of your screen.
Thanks, Nick, for giving us a lot of good information there. So we do have some questions that Nick and I can help answer looking through the question chat. We're also today joined by Dr. Albert Lee, who's Vice President of Chemistry here at EAG St. Louis. He's just here to help answer a few questions if there are any. So the first question that we have here is, Nick, you mentioned extractable, leachable, off-gassing, and in vitro testing could all be used to assess biocompatibility. Are all of these experiments necessary? If not, why pick one over the other?
Well, to answer that question, so, every product might have a different intended use and might have different risks associated with it. And depending on how that product is intended to be used, we might want to focus the testing that we do based on that. So if it's a wearable, extractable, leachable testing might be necessary, but if it's something that's worn near the user's face or could potentially give off something the user may inhale, then off-gassing may also be needed. And in vitro testing can also complement that as well.
That's all true. The only other thing I would maybe add, it may depend too on where the product is at in its life cycle and how quickly the results are needed to make decisions. We may pick in vitro testing to kind of get a very quick preliminary answer for something that's in more the developmental stages. But a product that's kind of just ready to go to manufacturing, we may want to do the full gamut of testing just to ensure that consumer safety. Next question. So here at Eurofins EAG, I looked at your website and saw that you actually do medical device testing as well as consumer product testing, which is what the webinar is about today. What's the timeline/cost difference for the consumer product testing compared to the ISO 10993 Part 18 medical device testing?
I'll take this one, Mary. So with medical device Part 18 testing, the goal is to identify all of the chemicals that are released during the extraction process. This ultimately goes to the FDA for approval or however else the data will end up being used. By comparison, the consumer product biocomp testing really focuses on chemicals of concern. In other words, we have a good idea of things that can be sensitizers or irritants that can come off of these consumer products. So it tends to be very targeted, as in we know what we're looking for, generally speaking, versus trying to identify everything else. So because of these differences between med device Part 18 testing, med device Part 18 does have a longer timeline and of course a higher cost because of it. And generally speaking, here in St. Louis, the Part 18 testing is completed with a 6 to 8 week timeline. The consumer product biocomp testing is roughly 3 to 4 weeks. So that's how they differ. The costs themselves, it would have to get down to the specifics for what are the products, devices that we're looking at in order to give a better number. But generally speaking, timeline at least, consumer products takes about half as long.
Have you had any experience responding to a CPSC complaint?
Yes, we have. We've helped our clients address complaints from the CPSC. We've even had people from our company testify for this Consumer Product Safety Commission. Right, Al?
Yeah, that's me. I had the pleasure of sitting before a panel and getting put on the hot seat. Our experience includes presenting the findings that we have in response to a complaint. We've also talked about the programs that we've worked with to provide chemical testing to support what's going on in the development cycle, as well as monitoring what comes out at the retail end. So yes, we've been before the CPSC.
In addition to that, we've also helped determine root cause analysis for the complaints themselves. So companies will come to us and say, you know, we've got a rush of complaints for the use of our product, and then we'll help identify things. That can include delamination, skin sensitization, things like that. A whole list of things that we can help with. That's kind of the investigative part that Nick discussed early on in the presentation. Nick, you mentioned LCMS, GCMS, ICP-MS, and NMR in your presentation. Is that the only 4 techniques EAG has capabilities in?
No, those are just the ones I highlighted, but we have a fully equipped lab that can support a lot of other different types of testing in St. Louis.
We've got one question here I see. How are your capabilities in understanding the presence of concentrations in bioactive substances, enzymes/proteins?
Since when we were working with a customer in the biocomp area, where it turns out they had an issue with contamination that was causing an allergic reaction. I think we ended up tracing it to the use of latex gloves in the process. And we ended up partnering with another laboratory to nail down the specifics of what those proteins were. That's how we eventually found out that it was latex. I think that's a very general answer to that question that was phrased. I don't know if you have anything to add to that.
Yeah, I mean, I think that covers most of it. I mean, if there's proteins that give off bioactive substances, we would be able to screen for those, but that may get sort of outside the scope of the typical biocompatibility testing we're talking about related to consumer products. As I mentioned with the investigative testing that we can perform, we definitely can support that kind of investigative work if it's a request that the client brings to us.
A couple more kind of more specific questions on the analysis. We've got one here. Can HRIPT be used instead of mass spec analysis to find out biocompatibility? HRIPT is an acronym. I believe it's a patch test that can be used. So, you know, I know that that's a common way to assess biocompatibility. I don't know that it could be substituted for a mass spec analysis. So the patch test would give you very generalized, is there a reaction or not? Whereas mass spec would give you the information to know what compounds are causing the irritation. In addition, it can also detail what parts might be causing the irritation itself, whereas a patch test would just be the general device on its own. So I'm not sure if that completely answers your question, but those are my thoughts. So mass spec would be much more specific than a patch test. Here's a question about faux leather that a lot of times they're latex based. Could that be a problem too? We actually have seen a lot of clients who've asked us to test for latex for that reason, because latex can be a real sensitizer or irritant for certain people if they're allergic to it. So most of our clients tend to steer away from using latex, and we have been asked to test for that in the past. And it said also, in addition to that, a follow-up question was, would rubber/silicone waterproofing be a problem? I think in order for us to answer that, we'd actually have to kind of bring that in as a project in our lab and kind of take a look at the specifics. And if you'd like to contact us, my contact information is here on here, and I'll get you to the right person to help you essentially scope out that work.
When you answered that question about the faux leather, it reminded me of some of the studies we did with leather compounds. That was some really interesting chemistry.
Yeah, there's a lot of tanning agents that can really come out of those things. Here's a question about your phenol case study, Nick. So how do you determine, it says, how do you determine what the limit for the phenol content in the product? So let me read the whole thing. Thank you for sharing about the phenol case with the breakdown in heat and using a UPLC to solve this issue. How do you determine what the limit for phenol content in the product is? So I'm not sure if that refers to kind of the acceptable limit or if it refers to kind of the limit of detection on the instrument. The acceptable limit in the product, so the kind of the threshold that would be above or below if it's safe for the consumer. We partner with third-party toxicologists to help us determine that. And that's how that's determined. I'll let Nick speak to kind of the limit of detection for the instrument.
Yeah, so I interpreted the question as being related to what's the level that's associated risk. And like Mary said, it would be up to toxicologists to evaluate that risk. We can only measure it and give the results. As far as for the instrumentation, the methods we've developed are very sensitive, so we can see, low digit ppm levels on instrument, and we determined this by comparison of all the data to try to see what we thought were the main culprits for the phenol we were seeing, and ultimately we ran standards to confirm which ones were degrading as well.
All right, great. Thank you so much, Nick and Mary and Al. Great job. And on behalf of EAG Laboratories and our presenters, thank you to everyone and have a wonderful day.